Welcome back. What I want to do in this video is talk briefly about a very, very important enzyme, and this enzyme is called lactate dehydrogenase. Lactate dehydrogenase. Now, before we really get into the function of this enzyme, I want to think about something. In glycolysis, we produce a lot of NADH, right? As we go through glycolysis, glyceraldehyde 3-phosphate dehydrogenase produces NADH. So let's say we've got a lot of NADH floating around, right? And the NADH comes from that enzyme, but also in the TCA cycle, it will come from isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase complex, malate dehydrogenase, and it will also come from the pyruvate dehydrogenase complex. So there's lots of enzymes that are producing NADH. Well, the ultimate goal of the NADH is to feed into the electron transport chain. And there's going to be an enzyme, and actually let me do it like this. There's going to be an enzyme in the respiratory chain, and it's and the enzyme is NADH ubiquinone, oxidoreductase. And what this enzyme is going to do is it's going to reoxidize NADH and turn it into NAD. But the thing you have to understand about this enzyme is it's completely dependent upon another enzyme down the chain. So this enzyme is often referred to as complex 1. But it turns out that complex 1 that oxidizes NADH is dependent on the activity of cytochrome C oxidase. Cytochrome C oxidase. And the general reaction of cytochrome C oxidase is it's going to take the electrons, and specifically 4 electrons, ultimately from NADH. Of course, there's a lot of intermediate steps, but it's ultimately going to take the electrons from NADH, and it's going to reduce oxygen down to water. Right? So the very oxygen that you breathe is the purpose of it is ultimately as a substrate for cytochrome C oxidase. Well, let's think about this. Cytochrome C oxidase is also known as complex 4, right? It's the last complex in the respiratory chain. So let's say this enzyme doesn't have oxygen. So let's say you're in an anaerobic environment. So this could be, say there's blockage of blood flow to a tissue, or maybe you're running as fast as you can and now your muscles are running on anaerobic metabolism. Well, what ends up happening is cytochrome C oxidase shuts down, and it's not due to some allosteric regulation. No, it's just due to the fact that it doesn't have its substrate. It doesn't have its substrate oxygen, right? So if cytochrome C oxidase shuts down, so too does NADH ubiquinone oxidoreductase, because essentially in order to power complex 1, there has to be a place for the electrons for NADH to go. And if cytochrome C oxidase shuts down, it stalls. There's no place for the electrons to go. And if that doesn't make sense to you, go watch the playlist on the respiratory chain. But ultimately, the electrons from NADH ultimately end up in oxygen. But if there's no oxygen, there's no place for the electrons in NADH to go. So NADH cannot be oxidized to NAD. So what happens? So NADH builds up. So NADH builds up, right? Well, if there's no place for NADH to go, then think about this. If NADH goes up, then NAD goes down, right? Because if you're not reoxidizing NADH, then the concentration of NAD decreases, right? Well, let's think about this. That enzyme in glycolysis, remember it was a glyceraldehyde 3-phosphate dehydrogenase, and that took G3P. It took G3P, and it converted G3P with the production of NADH. It turned it into 1,3-bisphosphoglycerate, right? And ultimately it required NAD, right? It required NAD. But think about this. If there's no NAD being regenerated, how is this enzyme supposed to function, right? Because if I'm not reoxidizing NADH in the respiratory chain, there's no regeneration of NAD. So that's why we have the enzyme lactate dehydrogenase. And so to understand this, let's look at the reaction of lactate dehydrogenase. It's going to look something like this. So you're going to start with pyruvate, right? At least in the direction that it's running now, it's going to start with pyruvate, right? And so what the ultimate goal of this enzyme is it's to regenerate NAD. And so it's going to take that NADH and it's going to transfer the electrons to pyruvate, and that's going to generate something called lactate. And so what you notice is the enzyme is named for the reverse reaction. And of course, this is an equilibrium reaction. It's named for the reverse reaction. So this right here is lactate, and you recognize the other one as pyruvate. So what do we see? What is the ultimate goal of this enzyme? The ultimate goal of this enzyme is to regenerate NAD, right? Because if NAD is decreasing, that means there's less of a substrate for glyceraldehyde 3-phosphate dehydrogenase, right? And if that doesn't function, then ultimately that portion of metabolism shuts down. There's no more pyruvate, right? Because if I'm not giving this enzyme NAD, then that enzyme doesn't function, and there's lower pyruvate, there's lower acetyl-CoA, and ultimately in general there's less energy production, and so if there's less energy production, the cell can die. So what ends up happening is lactate ends up being produced, and lactate is a waste product. It's a metabolic dead end, and in general, there's no place for it to go unless you can fix the problem. And if NAD starts to regenerate, right? If NAD starts to regenerate, then you start running glycolysis again, and in general, let me put it to you this way. Let's go back to our example of cytochrome C oxidase, the enzyme that required oxygen. Let's say that I have a lot of lactate present, right? Because I've been running anaerobically, right? I got a lot of lactate present, and this is the only way I'm regenerating NAD, right? Well, let's say I take a breather, I stop running or whatever. I stop running as fast as I can, and I catch a breather, and I'm getting more oxygen, right? Oxygen goes to my hemoglobin, oxygen goes to my tissues then, right? So I'm getting more oxygen. Well, then cytochrome C oxidase picks up again, right? Because now it has its substrate, and now NADH ubiquinone oxidoreductase picks up as well. So NADH continues to be oxidized back to NAD, and now my concentration of NAD increases. Well, if you think about it like this, right? In the reverse direction, NAD is getting reduced, is getting reduced to NADH, right? And so this is just Le Chatelier's principle, right? Assuming that I was running anaerobically, I probably have a lot of lactate present, right? And if I have a lot of, and likewise, I have a lot of lactate present, and if cytochrome C oxidase starts working again, therefore NADH ubiquinone oxidoreductase starts working again, and I'm building up NAD. And so by Le Chatelier's principle, it makes it more likely that pyruvate is going to form, right? So this is just an equilibrium reaction, and this right here, this is the reaction of lactate dehydrogenase. So again, to regroup, if I'm running on anaerobic metabolism, that means NADH is not being reoxidized to NAD+. And so that means that, and it's not just in glycolysis. There are other enzymes in the TCA cycle that require NAD. And if they're not getting NAD, then the whole metabolism shuts down. So the only way that the cell can regenerate NAD in the absence of NADH ubiquinone oxidoreductase is by this enzyme. So it's going to waste the NADH on pyruvate, and it's going to reduce it to lactate. And ultimately, lactate, like I mentioned, is a dead-end metabolite. There's really no place for it to go. At least in humans, there's no place for it to go. And so the only way you can really do anything with it is if you return it back to pyruvate. So once I start running aerobically again, and I start building up oxygen, cytochrome C oxidase says, hey, I got my substrate, right? So I'm going to start working. I'm going to start clearing those electrons, and then NADH ubiquinone oxidoreductase starts working again, and then I start regenerating NAD. And as NAD builds up more and more, I start to run this reaction in this direction. I start to regenerate NADH and regenerate pyruvate. And of course, then the pyruvate can then get consumed by the pyruvate dehydrogenase complex. And that also, too, is an NAD-dependent enzyme. So if you think about it, if I don't have any NAD present, that enzyme shuts down as well. So, and that's the enzyme that produces acetyl-CoA. So ultimately, running anaerobically is very, very bad, especially for prolonged periods of time. And to illustrate this point, a good example is a heart attack. And we also call a heart attack a myocardial because it's the muscle cells of the heart that are affected. Myo for muscle, myocardial infarction, a myocardial infarction. And essentially what this is when a muscle cell in the heart dies. Well, what would cause that? Well, let's say, first of all, on your heart, and I'm going to do my best to draw the heart. So on my heart, right? On the heart, there are coronary arteries, right? There's coronary arteries, right? And those are the arteries that are actually serving the actual muscle tissue of the heart, because the heart itself, we normally think of it as pumping blood to other areas of the body, but the heart itself actually requires blood as well because the muscle's contracting. Well, let's think about this, right? If I was to block this artery right there, then this whole area, right, that general area where that artery supplies, is not going to be receiving blood, and therefore it's not going to be receiving oxygen. So in that area, guess what? Cytochrome C oxidase shuts down because it no longer has its substrate. Therefore, the electrons aren't being cleared. So NADH ubiquinone oxidoreductase shuts down, and therefore NADH isn't being oxidized to NAD+, right? And so as a result, that cell is forced to use lactate dehydrogenase. And this is what we call anaerobic glycolysis. Let me write that down. Anaerobic metabolism, or anaerobic glycolysis. And anaerobic glycolysis, and actually we have another name for this, it's called fermentation, or lactic acid fermentation. That's essentially where we are converting pyruvate to lactate. And the whole purpose of it is carrying out, is so we can regenerate NAD and carry out glycolysis. But if there's no oxygen, then the respiratory chain doesn't work. And it turns out that heart cells cannot run on this for very long. And so because the heart is so dependent on aerobic metabolism that when it starts running on anaerobic for any period of time, the heart cell or the muscle cell dies. And that's what we call a myocardial infarction or a heart attack. And it's replaced by scar tissue, which cannot be returned back into muscle tissue. Okay? And so let's come over here and think about this lactate dehydrogenase, right? When I do lactate dehydrogenase, right, I'm going to be running on purely glycolysis, right? Because if there's no, if I'm not running cytochrome C oxidase, there's no ATP produced by the mitochondria. So normally what we can, and the number it changes depending, but let's say it's about 30 ATP produced by the mitochondria. So this is by the mitochondria, right? Versus 2 by ATP. And the net 2 is of course contributed by phosphoglycerate kinase and pyruvate kinase. And so this is by purely glycolysis. And let's say I asked you, which one is going to be easier for a heart cell to run on? And really, I could ask you which is easier for any cell to run on. And the answer that you would give me, green for good, is going to be 30 ATP. That's the easiest for the cell to run on. And you can imagine that the cell is going to be really stressed when it runs on 2 ATP. And that's actually why the heart cell dies. And in fact, it doesn't die in an apoptotic way. It actually dies in a very violent, uncontrolled way. And it's just a cell lysis. And so the whole purpose of lactate dehydrogenase is to regenerate NAD so that glycolysis can continue running. Because if the whole circumstance where you'd be where you'd be accumulating NADH is if the respiratory chain is shut down. So you'd be assuming that the cell is running anaerobically, and so you'd be only producing that 2 ATP, right? So I hope this video helped you get an intuitive sense of what lactate dehydrogenase is. And in other videos, we'll look at other various enzymes. See you soon.